from Microbe TV. This is a special immune booster recorded during the American Association of Immunologists conference in Chicago, May 2024. Starring Cindy Leifer and Brianne Barker. Featuring Luis Montaner from the Wistar Institute.
Welcome here today, and thank you for taking the time out of attending the conference to talk to us.
Thank you. Thank you for having me.
So we just wanted to start sort of at the beginning, however far back you want to go. How did you get interested in science? I know you have a slightly different career path than many of the other people we've talked to.
Yeah, I grew up in San Juan, Puerto Rico, and my father was a pediatrician, a family practice pediatrician. So I grew up with medicine being very much part of our environment and hanging out at his office and getting sort of immersed in that type of world of medical care. So I got intrigued into science from the very beginning, but I sort of wanted to do more than care. I was more intrigued in the research aspect and discovery. So I sort of as a child had all these sort of science kit stuff that you give your kid to kind of make a mess at the house. And then as I progressed, I then got very intrigued with veterinary medicine due to the multifaceted aspect of being able to take care of different systems, like, different physiologies. And I thought that was a lot more interesting. And of course, as a child, I would look at my father and say, well, that's just a human. So it's just one. And exactly. So I went to veterinary school with the intent to actually do research. So I remember in the interview process, sort of, they asked me, well, what do you want to do? And I said, well, I want to do research. And actually, no, it was a good thing. They were looking to diversify their ranks. So then I went ahead and did, instead of clinical rotations, I did, research rotations. And then I got an opportunity to work with simian immunodeficiency virus. And at that point, it was at the Harvard Primate Center at the time. And then I sort of started looking at what I was going to do next, and I knew I was going to do a doctorate in research. And so I was looking at multiple places, and I was fortunate enough to get a Marshall Scholarship, which is a scholarship offered by the UK government in sort of thanks of the Marshall Plan. And you basically get your education covered in anywhere in the UK that you would like to do. So I went to University of Oxford and worked on HIV and macrophage biology, and sort of finished my PhD at that point. And right as I was just finishing, I sort of met a sort of a pivotal person that would change my life. It was Giorgio Trinchieri at the time. We were at a meeting, and he said, well, why don't you come and visit Wistar Institute in Philadelphia and see if you're interested to come. And indeed, I was then recruited to Wistar as I was finishing my PhD. Under the premise that I had a doctorate with the veterinary degree before. But if you had seen me, this was like 27 years old. I looked like Harry Potter. This was definitely a vote of confidence on their part. And I basically developed my career at the Wistar Institute ever since. And we went from in vitro systems to patient-based research to translational, to animal models of non-human primates, to clinical trials in Philadelphia, clinical trials in South Africa. Now we have clinical trials in Vietnam, and starting to grow a global coalition of HIV cure research, both for understanding where the virus persists, also how to develop strategies that allow for durable suppression, or how to develop strategies for eradication. And all of this within the context of community engagement. You want to bring people living with HIV and advocates into the process so they can have an influence on not just learning what we're doing, but also helping inform what is a priority for them. So this trajectory has now really kind of culminated in the Martin Delaney Collaboratory Program, which we're one of 10 nationally, which is a large coalition of scientists both in the US and globally pursuing the HIV cure. And we're now in the second cycle of our current grant, and it's really exciting. We're making some progress.
Cool. So can you tell us a little bit about the types of immune responses that you're working on when you're thinking about working against HIV?
I mean, as you know, HIV has been able to surpass the pressure of the immune system, both from the T cell side and the B cell side, in order to kind of stay one step ahead of the commitment of the epitopes that the immune system goes after. And at the same time, kind of impaired the innate immune system to be dysregulated. So between these two, I mean, it allows it to persist. So it's kind of interesting that AIDS is not really observed earlier in the infection. If you think about how efficient the virus is in getting through these barriers. But nevertheless, in the context of the cure, we're building on the benefits of antiretroviral therapy. So we are expecting that current therapies will allow, and we know, recovery of the immune system. And as a consequence, now we have a choice. Okay, now if we have a recovered immune system, what type of immune response are we going to go after? And right now, there are several initiatives underway. From the T cell side, there is a heavy emphasis on chimeric antigen receptors, meaning how do we introduce new molecules that allow for more efficient detection or more efficient killing than the natural process of T cell commitment would allow for. There is less of an emphasis on vaccines for T cells because the whole jury's out as to how do you even develop a T cell response that's effective, as prefaced by the failure of the vaccine initiative and the efficiency by which the virus mutates out from selection. So CARs is a big emphasis on the T cell. From the B cell side, we're taking advantage of the broadly neutralizing antibodies. These are antibodies that have been identified in individuals that can neutralize virus across multiple persons. And that's quite exceptional because that's not the norm. So the concept is, can we take these antibodies, provide them with an effector strategy, or can they themselves provide some antibody-dependent cytotoxicity or some other effector mechanism that would allow for greater clearance in the presence of antiretroviral therapy. And the third is the innate immune system. I mean, this is where the natural killer cell becomes the lead effector cell that is being pursued in trying to determine, in similar manner as the T cell, can we empower the NK with chimeric antigen receptors that would make them work better? Can we remove negative checkpoints of the NK to be more efficient in providing sort of an antiviral response? And all of these three, whether it be CAR, NK, or antibody, are also overlaid with cytokine-based treatments, like, for example, IL-15 for the proliferation of effector T cells, IL-7, interferon, type 1 interferons for the antiviral effect. So among that portfolio, you could see that we have multiple strategies that are all kind of trying to maximize a response that would naturally not occur.
So are some of these also thinking about dealing with the latent reservoir?
That was exactly my question.
Yes, well, all of them. Because in the context of the antiretroviral therapy, the barrier for eradication is the persistent virus. And the persistent virus exists in two forms. One is a integrated defective form and an integrated replication competent form. And in the last 4 years, we've learned a lot more about how to measure the replication competent reservoir, and the approaches now that are not necessarily immunological but more virological is how best to manipulate that replication competent reservoir for either activation so that it could be expressed and then addressed by the immune system, or repression, block it and actually silence it as a strategy. So we've learned a lot more about the latent reservoir and how it persists in someone.
Very cool.
So do you think that your experience in coming up being a veterinarian, where I know because I'm out of vet school, where the thought process is much more broad than in human medicine, do you think that that's endowed you with some unique aspects to your research that has allowed you to move things forward?
I mean, of course, I'm going to defend the education. So, of course, veterinarians are wonderful and they are very critical thinkers. But I mean, I would like to think that yes, I mean, an education that kind of asks you to look at comparative biology, and it's also been very useful in the use of animal models, because we have employed and had reasons to use non-human primates in our studies, as well as mice, for some aspects of the biologies that we're trying to understand. So that kind of comfort to be able to engage animal models at the same time as sort of pursue human immunology and human clinical trials is been really useful. But you know, this is all collaborative work. So in a way, the fact that I have a veterinary degree doesn't really speak to coalition building, because in that sense, it's about bringing people together and identifying how best to work together. And in that respect, I've been fortunate that, you know, I've been able to develop the programs, and everyone that has contributed has seen the value of their contribution, you know. So because it's a lot of fun.
So perhaps just a question out of interest, what type of non-human primate model have you been looking at mostly?
Well, non-human primates are also different in regards to the types of
that's in fact why I'm asking.
Species that could be used. And so, but most of the work is in the rhesus macaque, due to the availability of a very predictable model of SIV infection, and more experience with how that model responds to both antiretroviral therapy as well as immunotherapy.
Cool.
And you mentioned this large coalition that you have. Do you want to say a little bit more about that and how you're running the trials in many different countries? Like, so what is a little bit more information about that?
So we have a website called beat-hiv.org. So please visit it, and you'll see all the descriptions of the research objectives and the community engagement. We have videos of prior lectures and all the components that we're engaged in as a group. I think the context of the group is divided into 4 major domains. One is understanding how the virus persists in a person living with HIV. So here's where the team's looking at the integrated replication competent and defective, or this is also where biomarker work is underway with both glycomic, metabolomic, or cytokine-based assays. Then the second part is strategies to allow for durable suppression. So this is where you're asking, can I convey a response that will control just as effectively as antiretroviral therapy, but not necessarily eradicate? And here's where strategies for antibody-mediated suppression or NK-mediated suppression have been sort of put DNA-based trikes and bites that allow for more effective control. And then the third is the strategies for eradication. And here's with gene therapy for CAR T-cell, lipid nanoparticle mediated delivery of CARs. But the durable suppression and the eradication kind of like merge with one another, because you could never, I mean, who could anticipate that you're going after durable suppression and then you spill over into eradication, or vice versa, that you do eradication and you spill over to. So nevertheless, this is how the group is organized. And then the fourth is community engagement, you know, and social science, this aspect of, do we have an understanding of how the community is accepting and responding to these strategies or these concepts, or how much and also providing a voice for them in regards to how these other research efforts are underway. The trials are kind of coming out of that group. Like, for example, we had a trial with testing 2 broadly neutralizing antibodies and interferon, type 1 interferon, as a strategy to reduce the reservoir, and that came from the group.
So those patient advocates are actively participating in deciding the directions of the research?
Well, they're contributing to the organization of the research, but they're not deciding per se the direction of the biological scientific questions that we're pursuing, but they are introducing components that should be part of the equation. I'll give you an example. We had an interest on a at-home viral load device in determining, because when participants are on trials and they stop antiretroviral therapy, they need to monitor for viral rebound very frequently, like weekly. And the question is, okay, well, if you're going to participate in a trial, you're going to have to come to the clinic every single week.
That's a lot.
So could we, which they requested, could we develop some strategy by which we could evaluate viral loads from home? And that allowed us to start working with Merck, and we tested, viral load devices that are currently, that's one of the projects that we're undertaking. Another one was, if we're going to recruit participants into trials, we can ask our community to go out and educate others and bring them. But we also then undertook a study with them to interview the providers, the medical providers, because it became evident that if the participant was going to consider a study, the provider needed to be approving.
Sure.
So then what is the level of awareness that the provider has on HIV cure research? And then we learned that we could do better in educating the medical community about what's actually the priorities of the cure research, and perhaps make any recruitment effort more efficient in the future. And that came from the community component.
Yeah.
And that's what I mean about kind of a synergy between the community contributing to the process, and then the science trying to bring that to them on a trial or for their general awareness.
So I think that's a really interesting point, because that community is going to bring a really different perspective, right? So, for example, like the home monitoring, we don't think really, these can just come to the clinic, but yet from their perspective, correct, that's an inconvenience. And if they voice that, then there's actually an advance that can be made in response to that. Okay. And so thinking about that whole diversity, which then brings me sort of my next question. So you're the chair of the Minority Affairs Committee for AAI, right?
For AAI here. For
yeah.
yes.
So can you talk a little bit about that? Because I think that's, your experiences, and then your involvement in all of this diversity and community engagement. How does that fit into everything? So describe MAC a little bit.
So MAC is the Minority Affairs Committee for the AAI, and its mission is to increase diversity, promote professional advancement among underrepresented groups. And the committee organizes sort of a professional development roundtable where there's like maybe 25 different tables, and there's faculty that come 2 per table, and there's like different topics like finding a postdoc or going into industry or government job, and the trainees can choose which one of the tables they want to be at, and they can sort of cycle through 2 tables over a period of an hour and a half. And we also highlight underrepresented scientists every year, and this year is Tanya Webb, who will have the awards lecture tomorrow, the Vanguard lecture.
And we have interviewed her, so if you're interested in finding that out, you can check the other episode of Immune out. Yes,
that's right. So anyway, she's wonderful.
Yeah.
And we also have hosted collections on the Journal of Immunology that highlight underrepresented scientists. And we are engaged throughout the year in trying to make sure that initiatives from the society and membership is well attended when it comes to underrepresented groups and providing equal opportunity.
That's awesome. Really important that you're doing that, and I'm so happy to hear about that. As we're also talking a little bit about the conference and being at AAI, can you tell us a little bit about some of the work that your lab is presenting here?
So my lab is presenting 2 stories this year. The first story is we successfully cloned antibodies that neutralize type 1 interferons, that 2 antibodies from MS patients that can neutralize beta but not alpha.
Oh, okay.
And then 2 antibodies from participants in our trials that were treated with interferon alpha that can neutralize alpha but not beta. And these sort of will be really important in teasing out the potential anergic effects of beta versus the antiviral effects of alpha, because a lot of the strategies up to now in the human have actually targeted the receptor, receptor 1, and that ablates all interferons. And so having human antibodies that were cloned from humans that could potentially give us the tools to tease between these two, I think will be really important. We already have data from work that we presented, not in this conference, but in the conference for opportunistic infections that occurred earlier in the year, that the use of, for example, the anti-interferon beta antibody, but not the interferon alpha antibody, could actually serve as a latency reversing agent in mice that are antiretroviral, and humanized mice that are under antiretroviral therapy, because there's an undercurrent of beta expression that persists even in the context of antiretroviral therapy. There have been also reports in cancer about immunotherapy being more effective with beta antibodies and alpha. So we believe that these would be a good premise for future work. The second story
I will say that story sounds amazing, and I can imagine so many places in viral immunology where those types of antibodies can be used.
If you look, there's no antibody. I mean, there are antibodies in the human. In the human, there's only 2 patents, one on alpha and one on beta. And if you try to buy these antibodies, we had to clone them from the patent sequence. They're not really available. So most of the reagents are available on the mouse, but not on the human. The second story is the outcome of the clinical trial for the use of the BNAbs, the 2 broadly neutralizing antibodies, and interferon, because a big surprise came on that study, which was, as you would anticipate, if you add interferon, you stimulate the natural killer cell. Yeah. And if you give antibodies, the natural killer cells will kill better. Fine. So, and it works beautiful in the lab, right? So we preface the same thing will happen in the humans. So we provided interferon in the same regimen as hepatitis C before the protease treatments, the weekly doses. We gave infusions of the 2 antibodies. But then what happened was that there was no change in the reservoir, and the NK response actually decreased for antibody-dependent cytotoxicity, but not direct cytotoxicity. Huh. And what happened was that there was a drop in the CD16 due to an increase in ADAM17. And the increase in ADAM17 corresponded to the loss of the ex vivo antibody-dependent cytotoxicity assay, as well as the expression of 16 in the 56 subset. But the activity against K562 cells, the direct cytotoxicity, did not decrease. So what we learned was that when we think about immunotherapy on NK-based strategies, we're going to have to rethink how we design studies, because it's not going to be like the T cell. In the T cell, you give doses, you stimulate the response, you expect the response to continue to get re-stimulated from the antigen and continue to work, and you only worry about exhaustion. Right. But on the antibody-dependent cytotoxicity of the NK, you have an exquisite early negative checkpoint on the cell itself, that as soon as you stimulate it, it would cleave. So that the more you give, the worse it will get, unless you let it recover before you do it, or you add an inhibitor against ADAM17. So that is the second story, meaning that, yes, we didn't achieve what we were after, but we learned something about the NK immunotherapy in humans that we did not know before.
That's really neat. And I know there's so much variability between human NK responses and NK responses in some other species. So it's important that you did that in human and actually could see that.
Of course, in hindsight, I'm sure that some old immunologists would say, well, of course.
If you had read the paper from 1945, you would have known this.
If you kind of like re-stimulate something, of course it's going to back, control itself in some way. But anyway, so we will move forward with that information.
Well, we could probably continue this conversation for a really long time, because this was really interesting, but I think we have to wrap it up now.
So thank you so much.
Thank you.
That's a special episode of Immune, an immune booster. If you have questions or comments, please send them to immune@microbe.tv. If you enjoy our work, please consider supporting the science shows of Microbe TV. Go to microbe.tv/contribute. Microbe TV is a 501c3, so your donations are U.S. federal tax deductible. Our guest today from the Wistar Institute, Luis Montaner. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Brianne Barker's at Drew University, Bio Prof. Barker on Blue Sky. Music on Immune Boosters is by Tatami. Thanks for listening to Immune, the podcast that's infectious.